Welcome to the Deep Dive, where we take source material and, well, really distill it into powerful insights. Today, we're embarking on a journey into the incredibly small. We're talking light microscopy and histology.
Yeah, it's a really fundamental topic, especially for you, our listener, stepping into your first year of medical studies. Our mission today is to equip you to not just understand these concepts, but to actually explain them clearly. Like, even to a curious 10-year-old, we'll try to build some strong mental anchors, some metaphors along the way.
Exactly. Think of this as your superpowered prep session, making sure you grasp not just the what, but the why it matters, and importantly, how to simplify it. We've got some excellent lectures, some good materials on this, so let's unlock these microscopic secrets. Okay, so when we talk about seeing tiny things, just how tiny are we really talking? Let's start there. With scale, what's the basic unit we're dealing with?
Understanding the scale is, yeah, it's absolutely crucial. We're moving way beyond what you can see with your naked eye. We're going from millimeters down to micrometer, and then even smaller to nanometers. So to give you that simple picture, if a millimeter is like a chocolate chip.
Okay, I like that. A chocolate chip.
Right. Then a micrometer is like a tiny sugar crystal on that chip. And a nanometer, that's even smaller, like an invisible speck of flavor in that sugar crystal.
Wow. The chocolate chip analogy really works. So we're talking thousand-fold jumps down each time, right? 1 millimeter is 1,000 micrometers,
Exactly.
and 1 micrometer is 1,000 nanometers.
Spot on. And most human cells, they actually hover around, say, 10 to 20 micrometers in diameter. So incredibly tiny. But what's really insightful is how the size range, it dictates our imaging challenges. It even tells us how cells do their jobs. You've got tiny red blood cells, about 7 micrometers, right up to an oocyte, one of the largest cells, around 100 micrometers. Each size tells a story.
That gives a great sense of just how small. But then, how good are our eyes for seeing these things? My own eye, it has limits, right? Things just blur together if they're too close. So what exactly is this limit of resolution? Why is it so critical here?
Yeah, the limit of resolution. Basically, it's the smallest distance where you can still tell two objects apart. Imagine trying to read, I don't know, really fine print from across the room. At some point, the letters just blur into one line, don't they?
Mm-hmm. Definitely happens.
Well, your eye has a resolution limit too, roughly 0.25 millimeters. If two tiny dots are closer than that, they just look like one blob.
Okay, so below that, our eyes just can't separate them. That's a huge limit when we're trying to look at cells, which are like way smaller than that. How do microscopes manage such a leap? How do they get better resolution?
Well, the practical limit for a standard light microscope, the kind you'll use a lot, is around 200 nanometers. And a big part of that is related to the wavelength of visible light itself. You can't easily resolve something that's much smaller than the light waves you're using to see it.
Okay, so light itself sets a limit.
It does, but an electron microscope, that completely smashes that barrier. It can resolve details down to about 0.1 nanometers. That's like 1,000 times finer.
1,000 times. Wow.
Yeah, it's a massive leap. And it's all because it uses electrons instead of light. And electrons have incredibly shorter wavelengths.
Okay, so that really is like having super goggles for the invisible world. So if the light microscope is our workhorse for seeing cells, nuclei, maybe mitochondria, what does the electron microscope let us see that's just impossible otherwise?
Well, with a light microscope, you see the whole cell, the nucleus, maybe those larger organelles like mitochondria, as you said, but really fine structures, like those tiny finger-like projections called microvilli. They're only about 90 nanometers across. They're just too small for a light microscope to really show clearly.
Right, it'd just be a blur.
Pretty much. That's where the electron microscope comes in. It lets us see individual ribosomes, those protein factories, which are around 25 nanometers, and those much finer details inside mitochondria, things completely out of reach for a standard light scope.
Okay, it's one thing to see these tiny things, but why do we care so much? What's the big picture value? Why look at tissue under a microscope in medicine?
This is where histology, the study of tissue, becomes just immensely powerful. Rudolf Virchow, often called the father of modern pathology, he famously said something like, each anatomical change must have been preceded by a chemical one. Histology helps us actually see those anatomical changes. It gives us visible evidence of those underlying shifts.
So it's not just curiosity then. You're saying it connects directly to patient care, life and death stuff.
Absolutely. Malignancies, cancers, for example, they have very characteristic histological changes, things you can see. The whole process of cancer staging, figuring out how advanced it is, which guides treatment, predicts prognosis, that relies heavily on what we see under the microscope.
Right. Without histology, we'd be flying blind in many cases.
Pretty much, yeah. Our diagnostic and prognostic abilities would be, well, severely limited.
You mentioned this is life and death information. Could you walk us through a scenario, maybe one where the microscope view really made the difference, something clinical observation just couldn't tell?
Sure, absolutely. Imagine a patient comes in, they've got an unusual looking mole. Clinically, just looking at it, it might be really hard to tell. Is it, an atypical but benign mole? Is it maybe a harmless seborrheic wart? Or is it something much more serious, like malignant melanoma?
And without the microscope, that could be a total guessing game, with potentially huge consequences for the patient.
Precisely. Histology gives us the definitive answer. We take a small tissue sample that's called a biopsy, and we look at it under the scope. We can actually see if there are dividing melanocytes invading the deeper layers of the skin, the dermis. We can measure something called the Breslow thickness, the depth of invasion. We can determine the specific type of melanoma. And all this direct visual evidence, it fundamentally impacts the treatment plan, the patient's chances. It's a really clear why for learning this.
That's a really powerful example. It makes the why crystal clear. Okay, so how do we get there? How do we get from that patient's mole or whatever tissue it is to that tiny prepared slide into the microscope? What's that whole journey look like? It sounds complicated.
It is a multi-step process, yeah. Almost like preserving a precious artifact, you could say. First, obviously, we need the tissue. That means doing a biopsy. This could be anything from a simple scrape, like a pap smear, small incision in the skin, maybe a needle biopsy for deeper organs like the liver or brain. The method really depends on where the tissue is.
Okay, step one, get the sample. Got it. But it's not ready to look at yet, right?
Right.
What's the next critical step?
No, not at all. The next absolutely crucial step is fixation. This is chemical preservation. It's designed to do two main things. First, stop the tissue from, self-digesting or decomposing autolysis and putrefaction.
Okay, stop it from rotting, basically.
Exactly. And second, it locks the cellular structures in place. It cross-links proteins and macromolecules. It's like hitting the pause button on decay, making sure the tissue doesn't change after it's removed from the body. We often use things like formaldehyde or glutaraldehyde for electron microscopy.
So fixation is like freezing the tissue in time chemically. What happens after it's preserved? It's still soft tissue, right?
It's fixed, but still wet and soft. So then comes embedding and processing. We need to get the water out first. So we dehydrate it, usually using increasing concentrations of alcohol, like 70% up to 100% ethanol.
Okay, swapping water for alcohol.
Yep. Then the alcohol is replaced with a solvent, something like xylene. This solvent mixes with wax. And finally, the tissue is infiltrated with molten paraffin wax, usually around 56 degrees Celsius. Once that wax cools and solidifies, it forms a firm block around our delicate sample. It's like encasing it in amber, makes it stable enough to slice really thin.
Amber. That's a great image. Freezing time carefully. But it makes me wonder, with all these steps, dehydration, solvents, hot wax, can things go wrong? Can the process itself introduce problems? Like, what's a common oops moment?
Oh, that's a really good question because, yes, precision is absolutely key and things can go wrong. That's why we always have to be looking out for artifacts. These are changes that, weren't actually there in the living tissue. They're introduced by the processing.
Okay, artifacts, like what kind of things?
Well, for example, tiny air bubbles can get trapped under the cover slip when you mount the section on the slide. Or if there's a tiny nick in the microtome knife, that's the super sharp blade used for cutting it, can cause scoring, like little scratch lines, or even tearing of the section. And these artifacts can definitely mislead you if you don't recognize them for what they are: processing errors, not actual biology.
Right, so you need to know what normal processing looks like to spot the mistakes. Okay, so once we have these perfect, super thin slices embedded in wax, they're still mostly transparent, right? How do we actually see the cells and structures?
Exactly. They're almost invisible. That's where staining comes in. Staining is what turns these transparent slices into, a vibrant roadmap of the body's internal workings, makes things visible. And the absolute workhorse, the most common stain by far, is H&E. That stands for Haematoxylin and Eosin. It uses two dyes.
H&E, the classic pink and purple. I think even I've seen pictures like that. So what does each dye actually do? What makes things pink or purple?
Okay, so haematoxylin is a basic dye. That means it likes to bind to acidic things in the cell. And what's acidic in the cell?
DNA.
In the nucleus.
Exactly. DNA in the nucleus, and also ribosomal RNA, which you find a lot of in the nucleolus and in ribosomes. So haematoxylin stains these acidic components a distinctive purple or blue color. We call these structures basophilic base-loving. Think of it painting the control center and the protein factories.
Okay, blue-purple for the nucleus and ribosome-rich areas. Got it. And the eosin, that must be the pink part.
Right. Eosin is an acidic dye, so naturally it stains basic components in the cell and outside the cell. This includes most proteins in the cytoplasm and also extracellular proteins like collagen fibers and connective tissue. It stains them various shades of pink. We call these components acidophilic or eosinophilic acid-loving or eosin-loving. So you put them together and say, in the lining of the stomach, you'd see distinct purple nuclei and pink cytoplasm in the cells. That's a fantastic visual anchor. You can immediately start to see the different compartments just by the colors.
Absolutely. And you can read more subtle stories too. Like the nucleus itself tells a tale. If the DNA is tightly packed, condensed, we call that heterochromatin, it stains intensely purple. That often means the cell isn't very active transcriptionally.
Okay. Dark purple nucleus equals quiet cell.
Pretty much. But if the DNA is unraveled, open euchromatin, ready for transcription, the nucleus looks paler, maybe with just the edges stained dark.
Yeah.
That signals an active cell. And in nerve cells, you see these clumps of purple staining in the cytoplasm called Nissl bodies. That's rough endoplasmic reticulum and free ribosomes, just churning out proteins.
Wow.
And if that nerve cell gets injured, those Nissl bodies can actually start to break down and disappear. It's called chromatolysis. It's a visible sign something's wrong.
So the staining isn't just color, it's revealing function, activity, even health or injury. Amazing. What about other stains? Is H&E the only game in town, or do we have special tools for specific features?
Oh, definitely not the only game. H&E is the standard overview, but we have lots of special stains. One really important one is the PAS reaction. That stands for Periodic Acid-Schiff. This isn't a single dye, it's a chemical reaction that specifically targets carbohydrates and glycoproteins, turning them a vibrant magenta color. It's like a special agent sent in to highlight just those molecules.
Magenta for carbs. Okay. What kinds of structures does PAS light up that H&E might miss?
It's excellent for visualizing things rich in carbohydrates, like the brush borders on intestinal cells. Those are packed with microvilli coated in glycoproteins. Also, basement membranes, which are sheets underlying epithelial cells, they stain well with PAS. And mucus produced by goblet cells, like in the gut or airways, stains bright magenta.
Okay, so it's great for boundaries and secretions. Any clinical relevance?
Yeah, absolutely. For example, in diagnosing certain kidney diseases, the changes in the basement membranes show up really well with PAS. And thinking about conditions like diabetes, where high glucose levels can affect glycoproteins in tissues, PAS staining can sometimes highlight those changes too.
So H&E is our general map. PAS is like a highlighter for carbs and mucus. Are there other ways to get even more specific, or maybe see things differently?
Yeah, lots of ways. Beyond PAS, there are tons of other special stains, like trichrome stains are great for distinguishing muscle, red from collagen, blue or green. Other stains pick out elastic fibers or reticular fibers or lipids. And then there's immunohistochemistry or IHC. This is really powerful.
Sounds like antibody.
Exactly. You use antibodies that are designed to stick to one specific protein target. And these antibodies have a tag on them, usually an enzyme that produces a colored product or sometimes a fluorescent molecule. So wherever that protein is in the tissue, the antibody sticks and you get a color signal right there. It lets you pinpoint the location and abundance of specific molecules, like finding estrogen receptors in breast cancer cells, which is crucial for treatment decisions.
Wow. Okay, so you can basically ask, where is protein X? And IHC gives you the visual answer.
Pretty much. And all these processes, H&E, special stains, IHC, they're all followed by quality control, QC, steps to make sure the results are accurate. And nowadays, increasingly, slides are scanned by high-throughput digital scanners, creating virtual slides you can view on a computer. Makes analysis and sharing much easier.
Digital slides. The future is here. Okay, we've talked a lot about staining and the standard light microscope. Are there other types of light microscopy that give us different views, different insights?
Oh, absolutely. The basic light microscope, what we call bright field, is just the start. The light microscope platform is really versatile. For instance, there's phase contrast microscopy. This is fantastic because it allows us to view living, unstained cells.
Living cells. Without staining. How does that work?
Well, it cleverly exploits the fact that different parts of a cell slow down light slightly differently. They have different refractive indices. Phase contrast optics convert these tiny invisible phase shifts into visible differences in brightness or contrast. So you can see organelles and cell structures quite clearly in living cells without using any dyes, which might harm them. It's great for watching cells move or divide.
That's amazing. Seeing life happen without killing it first. Okay, what else?
Then there's dark field microscopy. It's kind of the opposite of bright field. Instead of shining light straight through the specimen, it illuminates it from the sides obliquely. So the only light that actually enters the objective lens is the light that's scattered by the specimen itself. What you see is a bright image of the specimen glowing against a dark background.
Ah, like seeing dust motes in a sunbeam in a dark room.
Exactly like that. It's really good for making very thin or faint structures visible, things that might be hard to see in bright field. It's particularly useful for detecting certain bacteria, especially thin, spiral-shaped ones like Treponema pallidum, the bug that causes syphilis. They show up brightly against the dark background.
Okay, useful for specific bugs. And you mentioned confocal earlier. How is that different again?
Right, conofocal microscopy. This is a more advanced technique, usually using lasers for illumination. Its key trick is using pinholes to block out any out-of-focus light from above or below the exact plane you're looking at. So you get incredibly sharp, high-resolution images, optical sections, without the blur you normally get in thick specimens.
Eliminating the blur sounds powerful.
It is, and it's often combined with fluorescence. You can label different structures with different fluorescent dyes or tagged antibodies. Because you get these sharp optical sections, you can stack them up using a computer to reconstruct stunning, detailed 3D images of cells and tissues. Really lets you see spatial relationships.
Wow. Phase contrast for living cells. Dark field for faint things. Confocal for high-res 3D maps. Each one really is a different tool for a different job, isn't it?
Exactly, a whole toolbox just within light microscopy.
This has been just an incredible deep dive. We've gone from the scale of a sugar crystal to the complex art of staining and these different ways of seeing. So as our listener gets ready to actually sit down on a microscope, look at their first slides.
Yep.
What's your best practical advice? How should they start to actually read what they're seeing? How do they interpret that story on the slide?
Okay, yeah, practical tips. My biggest advice is develop a systematic approach. Don't just randomly zoom around. Have a routine. Practice is absolutely key to getting good at this. First rule, always start low, then go high. Start with the lowest power objective lens to get the overall lay of the land. See the whole tissue section first. Then, once you're oriented, increase the magnification to inspect the details. And importantly, reduce the power again when you move to a different area of the slide, just to reorient yourself.
Good advice. Resist the urge to immediately jump to 40x. Start broad, get context, then zoom in. Okay. What else? How do they make sense of the shapes and patterns?
Orientation is everything. You have to ask yourself, how is this tissue cut? How is it oriented? Is it a transverse section, meaning is it cut across a tube or structure, like slicing a cucumber? Or is it a longitudinal section cut along its length, like slicing a hot dog? Or maybe it's cut at an angle, an oblique section. Because the appearance of structures, especially things like glands or ducts or blood vessels, can look vastly different depending on the plane of section.
Can you give an example?
Sure. Think about a coiled sweat gland in the skin. It's like a long, twisted tube. If you cut it transversely, you'll see lots of little circles or ovals, cross-sections of the tube. If you cut it longitudinally along part of its length, you might see parallel lines, the walls of the tube. If you cut it obliquely, you get weird, elongated oval shapes. It's like slicing up a coiled garden hose, remember? Depending on how you slice it, you see totally different shapes, but it's all the same hose.
The garden hose. Perfect analogy for explaining that to someone. Okay, so check orientation. What else?
Also, try to distinguish between natural borders and man-made borders. Is that the actual edge of the tissue, or is it just where the biopsy knife cut through? That can be confusing sometimes. And then, critically, you have to constantly interpret the staining. Remember what the colors mean.
Right, back to the H&E clues. Remind us of some key interpretation points based on staining.
Okay, so a cell that's really busy making proteins, like a plasma cell making antibodies, will usually have a large, relatively pale nucleus, lots of that active euchromatin we talked about, and often a prominent nucleolus. An inactive cell, like maybe a small lymphocyte, will have a compact, very dark staining nucleus, mostly inactive heterochromatin. A dying cell often has a nucleus that's shrunken and intensely dark. We call that pinosis. So the nucleus tells you a lot.
Activity and health for the nucleus. What about the cytoplasm?
Right, the pink stuff. If the cytoplasm is very pink and maybe looks a bit granular, that often means lots of mitochondria think energy production, or maybe lots of secretory granules waiting to be released. If the cytoplasm has a diffuse purplish or bluish tint mixed in with the pink, that suggests lots of ribosomes and rough ER, indicating active protein synthesis happening right there in the cytoplasm. We call that cytoplasmic basophilia. And areas that don't stain much at all, that look clear or empty, that might be stored fat droplets, which get washed out during processing, or maybe mucus vacuoles.
So many clues packed into those colors and textures. It really is like learning a visual language.
It absolutely is. And by systematically looking, thinking about orientation, and interpreting those staining clues, you really do learn to read the microscopic stories these tissues are telling.
So from understanding that mind-boggling scale of cells, down to the nitty-gritty of fixing, embedding, slicing, staining, and finally interpreting, this deep dive has given us a really powerful foundation in light microscopy and histology. Hopefully you, our listener, now feel equipped not just to understand it, but maybe even to explain the basics to that curious 10-year-old using some of these analogies.
Absolutely. By connecting the why, its immense diagnostic power in medicine, the cancer staging, the mole example with the how, the detailed processing, the H&E, the PAS, the different scopes, we really hope you see that histology isn't just about memorizing pictures. It's about understanding the dynamic, living story that a tissue tells, even when it's fixed on a slide. It truly is a cornerstone of medical understanding and practice.
And maybe here's a final thought to sort of chew on. If Virchow was right, that every anatomical change we see under the microscope had a preceding chemical change, what might we learn by trying to imagine not just that static snapshot on the slide, but the dynamic, invisible chemical dance that led to that appearance? And maybe what chemical signals are still echoing in that fixed cell?
That's a great point. It connects the structure back to the underlying biochemistry and physiology. We really encourage you to keep exploring, keep asking those why questions, and remember, practice, practice, practice is the only way to truly master the art of reading these microscopic stories. Get that scope time in.
Couldn't agree more. That's all for this deep dive. Until next time, keep learning, keep questioning, and keep exploring the incredible world around us, and maybe especially the world within us.